Hello and welcome to this video in the series on integrated disease management in cereals. And in this video we're going to consider how long is it that we need to protect the crop canopy for. In the previous video we looked at the question of which parts of the crop canopy need protecting and when. We understood something about the physiology of how yield forms and how disease impacts on that yield formation and therefore when the crop needs protecting. On this occasion we're going to think a bit more about how long do we keep that protection going for. Do we need to protect the green canopy right through to the last drop, or does there come a certain point when we can be a bit more relaxed? So let's get started. This is a video on the science and practice of integrated pest management focused on disease control in cereals. We're focusing in this video on the question, how long does the crop canopy need protecting? Because it's a principle of IPM that we only protect the crop when it needs it. So to be able to answer that question, we need a method of measuring canopy duration in order to be able to measure the effect of canopy duration on yield. And in this series of slides, we're going to explore some of the theory and evidence about the effects of green canopy duration. So there are two methods of measuring canopy duration that are commonly used. One we can describe by going back to a chart shown in a previous video where we looked at the effect of disease on the amount of green area index in the crop canopy through time, and we saw how disease reduced the green area index of the canopy during the later stages of crop growth. So the first method of measuring canopy duration is called healthy area duration, or HAD, and we can measure that by plotting green area index through time and integrating the area underneath the curve, which is a fancy way of saying that we add up the area underneath the curve, which in this case is shaded in yellow. And we can do that over the period of growth that's relevant to grain filling, for example. The other method we have is a bit more complicated. We could, for each day, measure how much green area index we've got. We could then, through Beer's law, which we looked at in a previous video, look at how much of that day's solar radiation would be intercepted by a canopy of that size. And on that day, we could measure the amount of sunshine that was available to the crop. And that would allow us to calculate exactly how many megajoules of photosynthetically active radiation the crop would have intercepted on that particular day. And then we could add up those amounts over a period of time. And that would give us a measure which is known as healthy area absorption, or HAR. So some years ago we did this for a series of field experimental plots. We did it over 2 years. The plots were very heavily affected by a yellow rust epidemic, and some of the plots were protected by fungicide treatment, others were left untreated, and some were only protected by low levels of fungicide input. So we got quite a big stretch of the amount of HAD and HAR that we measured in the crop. So in the chart on the left, we have the amount of HAD on the bottom axis and the amount of grain yield that we measured up the vertical axis. We can see that in plots that were very well protected against the epidemic, we had over 350 units of HAD. And this was measured during the period when stem reserves were being accumulated, and then during the period of grain filling through to senescence. In plots that were not protected at all against the disease, we lost virtually the entire canopy, and we were down to about 100 units of had. And you can see that the relationship between had and yield is curved, and that's a result of the fact that the size of the crop canopy at any point in time is related to the amount of light interception through Beer's law, which we looked at in a previous video, which is also curved. On the right hand side, in contrast, if we account for that curvature in Beer's law and count the number of megajoules of light energy intercepted and plot that for exactly the same field plots in exactly the same years, we see that we get a nice straight line. In some cases for wheat, we have seen the top of this line start to plateau, but in the vast majority of cases where we've measured this, what we find is that there's a straight line relationship. The more healthy area absorption we have during that grain filling period, the higher the wheat yield. Okay, so now let's consider what the processes are that link through between disease control and yield, and we'll do that for wheat and barley in order to look at the contrasts between them. So if we look at the series of charts on the left hand side for wheat, and we start at the bottom right. So along the bottom there we have the amount of disease control input that's made to the crop. Now that can be any type of disease control input. It could be the number of disease resistance genes that a plant breeder puts into a variety. It could be the amount of fungicide treatment that's applied, or it could be any number of integrated control measures. And the more we control the disease, the more green area index we're going to have, which is up the vertical axis of that bottom chart. But it's a law of diminishing returns because we can't have less disease than none. And therefore, as we increase the disease control input, we're kind of gradually approaching complete control of the disease, but we never quite get there. So disease control relates to green area index through a curve. And then if we move to the next chart up, we see that green area index relates to the amount of light we intercept through the Beer's law relationship, which again we looked at in previous video, and which is also curved. If we then look at the next chart up, we see that light interception is related to total dry matter produced, and that's a straight line, and the slope of that line represents the radiation use efficiency of the crop, which is the number of grams of dry matter produced per megajoule, and that's determined largely by plant breeding and through nutrition. And then we move to the final chart at the top, which shows how the total dry matter accumulated during this period of grain filling relates to grain yield. And we can see again that's a straight line, but it has an intercept at the left hand side. So even if we accumulated no dry matter during grain filling, we would still have some yield because of the translocation of stem reserves which have been accumulated before flowering and which are moved to the grain during grain filling as a kind of buffer against poor conditions during grain filling. That then allows us to look at the relationship between the disease control input and yield. So looking at the bottom axis and the top axis of our series of charts, if we plot those in the chart top right, we can see that our disease control input relates to yield through a curve because of the curvature of our effective disease control on GAI, and because of the curvature of Beer's law. And that means there will typically be an economic optimum amount of disease control input beyond which the costs of additional input are not paid for by additional yield because we're approaching the plateau. If we then turn our attention to barley, which, as we discussed in a previous video, behaves quite differently. So again, we have the disease control input at the bottom affecting green area index, but then the effect of that green area index that we're primarily interested in is what that does to light interception in the period before flowering, when sink capacity is being determined through tiller production and survival and through floret production and survival. And what we can see in the next chart up is that that light interception pre-anthesis before flowering pretty much linearly determines the number of grains per metre squared that we get, and the number of grains per metre squared that we get pretty much determines the yield that we obtain. And this is because barley is predominantly limited by the sink capacity for storage of carbohydrate in grains. And if we look what that relationship between light interception and grains and yield looks like in real data, this is data from the AHDB Barley Growth Guide across a range of sites and seasons from the field. And what we can see is if we measure the number of grains per metre squared, which is determined by light interception predominantly, and the grain yield of the crop, the two are very tightly related. So in barley, high yields are dependent on sufficient numbers of ears and fertile spikelets per ear. The aim is to control disease to maximise growth during tillering and tiller survival and as ears form, and there's limited scope for an increase in grains per ear to compensate for low numbers of ears per metre squared. So the question then is, well, how long do we need to keep this canopy alive for in barley? How long do we need to protect it against the effects of disease? And we looked at that in a series of experiments that were conducted in England and Scotland over a number of seasons. The ideal way of working out how long we need to protect against disease in barley would be to have a series of well-behaved epidemics that stopped and started when we wanted, and we could measure their effect. But of course, disease doesn't really behave like that. So we had to find a surrogate for disease that would mimic the effect of disease on the amount of health area absorption that the canopy was obtaining from solar radiation. So I'll just explain the experimental setup before we have a look at the results. So what we applied to different plots were a series of shades that reduced the amount of sunshine available to the canopy by about 2/3. So it was kind of equivalent to a pretty severe epidemic, reducing the green canopy and reducing the amount of light interception. And the chart on the left explains the treatment. So along the bottom we have the number of weeks after the ears were half emerged, so growth stage 55. So there was one set of plots that were completely unshaded throughout, and all of these plots were very well protected against disease, so they remained healthy for as long as possible. The next line up above the unshaded represents some plots which were shaded for the last 2 weeks of canopy life. So from about 6 weeks after ear emergence onwards, they were shaded. The next line up was a series of plots that were shaded for the last 3 weeks of their life, and then going up for for the last 4 weeks, the last 5 weeks, the last 6 weeks, and so on, until finally we had a series of plots that were shaded all the way through from the point at which the ears emerged. And we were then able to measure the effect of that on yield. So in the chart on the left, we have the point in time at which the shading was applied in weeks after ear emergence, and up the vertical axis we have the yield in tonnes per hectare that we measured. So the solid black dots with the solid black line represent the yield data, and we can see if we start bottom left of that line, not surprisingly, if we shade the crop and reduce photosynthesis by 2/3 throughout the period after emergence, the yield is substantially reduced and we only got about 2 tonnes per hectare. If we delayed the onset of shading successively by starting it a week after the ears were emerged, 2 weeks, 3 weeks, 4 weeks, 5 weeks, we can see the yield increases. But then we reach a point where the crop is insensitive to the reduction in light energy absorption, measured as HAR, and the yield plateaus. So essentially at that point or beyond that point, the crop is limited by its sink capacity, and having more photosynthesis for longer is not adding to yield because there's nowhere to put the carbohydrate. So we repeated this across a range of sites and seasons. We got a very similar pattern in all cases, and what we found was that it was critical to protect the canopy and light interception for between 3 and 5 weeks after the ears are 50% emerged. And at that point of time, in a very well protected crop, we would typically have about half of the canopy still green. So if we look at the dotted line, which is green leaf area, so we've got about 50% of the canopy green still, but beyond that point, it's not actually adding to yield. So what can we conclude from all of this? So how long does the crop canopy need protecting? So we have a method for measuring the duration of crop canopy through HAD and through HAR. We've shown that HAR relates more consistently to dry matter accumulation and yield than HAD, but it's more complicated to measure. But we can use these measures experimentally. For wheat, what we find is that no further grain filling occurs once grain moisture is below about 45%, which usually occurs around growth stage 87, and it's possible to maintain some green canopy beyond that point without benefit to yield. So we sometimes see crops with green stems to some extent really quite close through to harvest. However, in general, the higher the health area absorption, the higher the yield, because wheat usually has sufficient sink capacity to store the extra dry matter that's produced. And we're really only limited in the duration of canopy that we're trying to achieve by the economics of the costs of control. So the economic optimum for canopy size and duration will be a bit below the maximum achievable due to the costs of nutrition and crop protection. In spring barley, which was the crop in which we did this work, although we believe winter barley will behave broadly similarly, it's critical to maintain green canopy for at least 3 to 5 weeks after the ears are half emerged, and further increases in healthy air absorption beyond that may not increase yield as sink capacity is likely already to have been filled. So we're not saying that we can ignore late epidemics of Rinco, net blotch, ramularia, but we are saying it's not necessary to hang on to the last dying stages of green canopy, because we'll often be limited in any case by sink capacity. So thank you very much for watching.